Okay, so now for something completely different. I'm going to talk to you about genotoxicity biomarkers that we use in Plymouth. So essentially, a lot of the work that I look at focuses on damage to DNA that's been caused by some contaminant or other. And the work that we do tends to be contaminant specific, but these tests and tools can be applied to practically any contaminant at all. So hopefully they have fairly wide-reaching uses. They are both lab and field-based as well, so although we don't have a lot of field data at the moment for some of these tests, they are widely used in human biomonitoring for chemical effects, and there is the potential to actually look deeper into any issues that you might have in one of your catchments. If there's an area where there is an ecological problem and perhaps you don't know why, we can start at the very sort of basis at the DNA and work upwards and maybe answer the question why, as opposed to where is it happening and what's happening. So the reason that we look at DNA is because obviously it's the basis for everything. So what I wanted to try and emphasise with this slide is the fact that even though it sounds very abstract that we're looking at damage to DNA in an organism, that actually it's really linked to all these different effects at different levels, and these are the ones that you'll probably be incorporating into your models so far, so ecological effects, things that happen to populations. But what we find is that actually all these are underpinned by changes here, and all of these are underpinned by changes here to a degree or another. So once we look at damage to the DNA, we can actually start to investigate early on what these effects might be, or whether or not you're seeing a trend, a decrease in quality of a population or not. Like I said, these are related to human biomonitoring. So the top slides, I'm just trying to remember that. The bottom slides are human tissues. The top slides are tissues from a mussel, marine mussels. So you can see that they're very similar. This is a highly conserved process. So in terms of ecosystem services, if you look at the DNA damage in these organisms, you can also start thinking about potential effects on the human population as well. I also wanted to highlight that although a lot of the work that I do is work on marine mussels, these tests have been validated in a wide range of aquatic organisms, ranging from saltwater up to freshwater, to tropical fish, to invertebrates, of a whole variety of different species. So that means that these tests are also applicable to a wide range of ecological scenarios, depending on where you are in your catchment, or in our case, where you are in Plymouth Sound. Okay, I'm not going to go into this in too much detail, but I just wanted to mention a couple of the tests we use in case their names come up so they'd be familiar to you. This is actually a very simple process, doesn't look like it. It's also a very cheap process that just requires a few chemicals. So this test can be used in the field, on field-based samples, and in the lab as well. And it's essentially a measure of DNA damage. This cell here has not been damaged at all, as from a very healthy looking mussel. And what we do is we look at the percentage of DNA that's actually been broken. So you can see here that there's not very much intact DNA, there's absolutely shed loads of damage. So that indicates that this mussel was having a hard time of it. And these effects can be correlated, like I said earlier, to things higher up, which might be of more relevance to you guys. But this effect would be seen much quicker than a change in population. So it's kind of an early warning signal. We also use a test called the micronucleus assay. This is standard practice in human biomonitoring, so it's regulated. If a chemical is to be used for human consumption or in cosmetics or drugs, it will have to go through this process at some point to check whether or not it damages DNA. Essentially, a cell divides, and if there's a break in the DNA, mitosis, you can get a second nucleus in the cell, which indicates that this sort of process, some sort of radiation or chemical, has actually caused this sort of damage. And the amount of these in the cell can be diagnostic of certain types of chemicals as well, and particularly the difference between radioactive stuff and chemical stuff. Okay, so to summarise, these tests are sensitive, they're really sensitive, which means they can act as early warning signals for higher level effects. They have various equivalents in human biomonitoring, which obviously has links between us and the ecosystem. They're widely ranging in terms of species, and that means applicability. I've already said they're linked to higher level responses, and we also have a lot of other techniques that go through those levels as well. So if you do have any sort of areas where something's happening and you're not sure why, we can use these techniques to start to pick that apart, essentially. Thank you very much.
Brilliant.